If this presentation doesn't knock your socks off, I don't know what will. If you're searching for some sort of science-based insight into dementia for yourself or a loved one, then you come to the right place. As the former head of medical education for a microbiome firm, and now on my own, I have launched my educational platform for people just like yourself. People who are frustrated with the system as it is. I take you on this journey from the perspective of an expert in the amazing and rapidly evolving field of the microbiome. When I use the term microbiome, most of the time in my presentations, I'm referring to that of the colon. Although the microbiome of the entire gastrointestinal tract has relevance to include the mouth. But you may ask, how does the balance of bacteria and other actors in my mouth or colon have anything to do with dementia? Well, buckle up and open up your mind. Let's start by piquing your interest with a couple of amazing case studies, showing you how dementia can be reversed by switching microbiomes. That's right, it's possible. These highlight the power of the microbiome, and as we progress in this presentation, I'll explain to you how this is even possible. In this case study, an 82-year-old man was being treated for recurrent C. diff infection. He also suffered from dementia. The patient's wife reported that he no longer appeared to enjoy socializing and required considerable assistance with basic tasks such as food preparation, bathing, and taking his medication. Neuropsychiatric testing revealed significant impairments in memory and other areas. Following fecal microbiota transplant for his C. diff infection, at the follow-up visit 2 months post-FMT, the patient's wife reported improvements in the patient's mental acuity and affect. Testing indicated normal cognition. 4 months post-FMT, the patient reported continued improvement in memory with no progression in symptoms. The patient now remembered his daughter's birthday, which he had not been able to recall previously, and was able to correct the physician's recollections of his symptoms. 6 months post-FMT, the patient reported a marked improvement in mood and was more interactive. Here we have another FMT for C. diff, this time for a 90-year-old woman with Alzheimer's dementia. She had experienced a gradual decline in memory and cognition over the previous 5 years. She no longer appeared to enjoy socializing and sometimes required assistance with basic tasks. Cognitive tests revealed significant impairments in a number of areas. Upon failure to treat her C. diff, she underwent an FMT procedure. Following the first FMT, her severe gastrointestinal symptoms improved and a stool test for CDI was negative. 1 month after the first FMT, her cognitive function slightly improved. 3 months after the first FMT, she was diagnosed with a recurrent C. diff infection and she underwent a second FMT. 1 week after the second FMT, her severe gastrointestinal discomfort improved with negative CDI stool results. She reported a marked improvement in mood and daily living activities and showed more expressive affection. Figure 1 shows her improvements in cognitive function. Now that I have your attention, let's back up and do an intro. Globally, there are some 44 million people with dementia, and it's the 6th leading cause of death in the United States. There are various types of dementia, but we'll focus on the 2 most prominent ones, Alzheimer's, which comprises some 70 plus or minus % of all dementia, and dementia with Lewy bodies at some 20 plus or minus %. More than 95% of Alzheimer's cases are late onset, which is heavily influenced by environmental factors. One of those factors is metabolic syndrome, think type 2 diabetes. Although the correlation and connection here is strong, we're not going to dive into this, what's called type 3 diabetes. We'll focus on the microbiome component, which in my opinion is much stronger. And besides, everything's connected. Metabolic syndrome and all facets of your health are in large function a part of your microbiome. As for the genetic connection to Alzheimer's early in life, there are mutations in genes for encoding proteins for amyloid beta, but more on that later. Another genetic risk factor for the late onset cases, and there are 3 well-described major isoforms called ApoE, apolipoprotein, designated as 2, 5 to 10%, 3, 65 to 70% of us, and 4, 15 to 20%. ApoE4 is a major genetic risk factor for Alzheimer's, increasing it 15-fold in homozygotes, which is genes from both mom and dad. whereas ApoE2 reduces Alzheimer's by almost half. So let's focus on this ApoE for a bit to help us understand the origins of dementia. Apolipoprotein, as the name implies, is a protein which transports lipids from A to B. In the periphery, it's mainly produced in the liver and transports cholesterol and other lipids. In the brain, ApoE does the same, but to neurons. In the CNS, central nervous system, ApoE-mediated cholesterol and lipid transport plays a critical role in synapse formation and tissue repair. It is drastically upregulated in activated microglia and in stressed neurons under injury and pathological conditions. Like what? How about infection, say a bacteria? ApoE plays an important role in innate immunity, modulating immune neural inflammation in response to infection. ApoE turns on your own antimicrobial proteins. It is well established that ApoE co-deposits with amyloid beta and amyloid plaques. The interaction between ApoE and pathological amyloid beta deposition appears to be a central mechanism by which ApoE contributes to Alzheimer's risk. In addition to promoting amyloid beta plaque formation, ApoE is also involved in the clearance of amyloid beta deposition via various mechanisms, and this uptake process is impaired in the ApoE4 carriers. So basically, it plays a large role in neuronal antibacterial defense, tissue repair, and cleanup after the battle. So if you have a genetic flaw in this protein, then you can see how you're less able to fight off a bacterium, repair any damage from the ensuing inflammation, and clear out the debris after the fact. So let's see what happens in real life. I've covered LPS lipopolysaccharide in my presentations multiple times. LPS, aka endotoxin, makes up a large part of the outer layer of gram-negative bacteria. That's usually, but not always, where the bad guys reside. Our immune systems have evolved to strongly key in on LPS because it's a sign of invader and requires the appropriate alarm system to mobilize the troops. Insofar as the relationship between LPS and ApoE4 is concerned, we can see here when LPS was injected into the subjects, the inflammatory response for those with the E4 was much higher for those with the E3, which is the normal common version, as shown here in Figure 4. One measure is shown in Figure B, that of TNF-alpha. TNF-alpha is a key member of the inflammatory response. In fact, it's so key that there are a slew of drugs in the marketplace to inhibit its activity, which of course is not treating the root cause, but it makes a lot of money. As I frequently say, inflammation plays a key role in just about every condition that you can think of in the body. And of course, it includes dementia. In this study of subjects with mild to severe Alzheimer's disease, their cognition and TNF-alpha was assessed in conjunction with what they called SIEs, systemic inflammatory events. As you can see in Figure 2, cognitive function dramatically worsened following a high TNF-alpha response to an SIE, like we just saw with the ApoE4 versus the ApoE3. And what were these SIEs? These here in Table 1. The worst events that impacted dementia the most were, in ascending order, other infections, genitourinary infections, and gastrointestinal infections, as in bacteria, as in LPS. In fact, Alzheimer's plasma LPS levels are equal to those with liver cirrhosis. Well, that's not good. And just below HIV/AIDS, that's also bad. Now, like I said, LPS comes from gram-negative bacteria, which is where most of the bad actors reside. But not all LPS is created equal due to their structures. For example, the LPS from various species of the genus Bacteroides have far lower, if any, in the case of B. dorii, immunostimulatory nature than the LPS from E. coli. So you don't want a lot of E. coli circulating in your body. Where does the E. coli come from? Largely from the gut. When you have dysbiosis, E. coli is a classic opportunistic pathogen. Almost every single one of us has detectable E. coli in our guts. It's normal. And we also have LPS in our serum, also normal. The question is, what's the balance? And are you healthy enough to clear it? In a healthy microbiome, the good guys rule the day. But in dysbiosis, the bad guys take charge, and many ugly things happen, one of which is gut permeability. When large quantities of whole or parts of bacteria enter circulation, they can wind up anywhere. Mean blood endotoxin/LPS levels are increased threefold in Alzheimer's patients, and brain endotoxin levels are increased 2 to 3-fold. And endotoxin is also found in Alzheimer's amyloid plaques. And it's not just LPS, but other parts of these bacteria. Staying on the theme of E. coli, we see in this study, which looked at the presence of E. coli DNA in the white and gray matter of the brains of deceased healthy controls as compared to Alzheimer's disease subjects. In Figure C, in both white and gray brain matter, the Alzheimer's patients had significantly higher levels of E. coli DNA in their brains than did the aged matched healthy controls. Again, we're talking pro-inflammatory responses of the immune system to types and amounts of bacteria where they don't belong. We just looked at E. coli protein in the brain. Now we'll look at LPS. Here you can see how the LPS levels in the brain were significantly higher in Alzheimer's subjects as compared to controls. These are average numbers. In some samples from advanced cases, there were 26 times the level of LPS versus the age-matched controls. Now let's see this all in action. In this additional study where LPS was injected into subjects, a brain PET scan was done to measure the response. The PET scan tracer binds to a molecule upregulated by microglial activation, basically the neuroinflammatory response. So brain inflammation. The microglia are the primary immune surveillance cells of the brain. Microglia are necessary for the removal of damaged cells and promotion of tissue repair. However, inflammation, prolonged or acute, interrupts normal microglial function, leading to neuronal dysfunction, neurotoxicity, and neurodegeneration. Now here, the E. coli LPS was injected into the body, not the brain obviously. But look at the effects noted in the color C. Baseline reading is all green, while the post-LPS colors are largely orange, signaling microglial activation, or alarm, invader. The resulting systemic symptoms were a significant rise in TNF-alpha, like we saw earlier, as well as fatigue, headache, muscle pain, and shivering, all showing their normal response to this acute signature of infection. But we're not just concerned about acute inflammatory responses to bacterial signals as seen here, and with the elderly who got infections earlier, but we're equally concerned about long-term, low-grade infections lasting years. And this long or short-term exposure doesn't necessarily have to come from the gut. In fact, the mouth, which is another huge reservoir of bacteria, has also been shown to be highly associated with dementia. In a study using a huge database of over 30 million subjects with normal oral health versus poor oral health, the researchers found that there was a greater than 2-fold increase in Alzheimer's in the poor oral health cohort. Diseases related to tooth loss were the most significant risk factor for Alzheimer's. There are several other reports suggesting tooth loss increases the risk for dementia. And there are many more studies highlighting the connection between the oral microbiome and dementia. Whether from the gut or the mouth, the opportunistic pathogens will do bad things on their home turf and beyond. The prime opportunistic pathogen of the mouth is Porphyromonas gingivalis. Given the existence of P. gingivalis LPS in Alzheimer's disease brain autopsy, the following researchers took a closer look at the connection. P. gingivalis is another gram-negative bacterium that produces major virulence factors known as gingipains, which are proteases. They tear apart proteins. In C and D, they're identified as KGP and RGPB. These enzymes are essential for P. gingivalis survival and pathogenicity, playing critical roles in host colonization, inactivation of host defenses, iron and nutrient acquisition, and tissue destruction. These researchers found that when looking at the brain, the Alzheimer's subjects had significantly more of this toxic protease as compared to controls. Doesn't that sound like a recipe for inflammatory disaster? How do they get there? Well, if you have dysbiosis in your mouth, and then you brush or floss or even eat, plenty of studies show oral bacteria moving to other sites, to include the brain. So the nasty little features of protein degradation, which makes P. gingivalis so troublesome in the mouth, one can only assume is more so in the brain. And this study helps support the previous. Their rationale for the research states that, quote, multiple studies have shown up to a 7-fold higher density of oral bacteria in Alzheimer's brain tissues compared to normal. In Figure D, you can see the results, with the Alzheimer's brain samples having 5 to 10-fold more bacterial DNA reads versus controls. These listed here are phyla, so we have no idea where these bacteria come from. Perhaps some from the mouth and the majority from the gut. With the gut in mind, we now switch gears to that all-important region. In this study, 89 subjects from normal to dementia underwent a PET scan and blood analysis. Again, we're looking at microglial activation, like in the other PET study, and how that relates to markers in the blood. Not surprisingly, they showed that neurological inflammation was, in every case, correlated to LPS and butyrate. The more LPS, the higher the inflammatory signal. The more butyrate, the lower the inflammatory signal. Now, we already know what the LPS is doing. How about butyrate? If you've been watching my videos, you should have an appreciation for this amazing compound. For much more information, see my video dedicated to it. Bottom line, it is one of the most important health-promoting compounds in the body. And the most beneficial bacteria in the microbiome are all butyrate producers. And together, they help maintain gut integrity by limiting bacterial passage into general circulation. So let's take a look at a fecal microbiome analysis in dementia. Here, the gut microbiota was analyzed in 28 patients with dementia with Lewy bodies and 224 patients with Parkinson's disease. We'll touch on Parkinson's more in a few slides, but for now, just know that there are similarities between Parkinson's and dementia. But here, we're comparing the gut microbiomes of healthy controls to those with Lewy body dementia. As we can see, many of our usual suspects are shown. The healthy controls had significantly more classic health promoters, such as E. rectale, Agathobacter, Coprococcus, Faecalibacterium, and Fusicatenibacter, while the dementia subjects had significantly more bad actors like Agarthella and Aratorchis. When I compile all of the data from the 24 studies I could find from my meta-analysis for human fecal microbiome studies in dementia, I get the following results. This is the fecal microbial fingerprint for dementia. As you can see, once again, the healthy controls consistently had significantly more classic health promoters like E. rectale, F. Troussetii, Roseburia, and Ruminococcus in their microbiomes as compared to dementia subjects, as denoted in green, whereas the dementia subjects had significantly more species from Enterobacteriaceae, which is the worst family in the GI tract, which is usually represented by our good friend E. coli. Also, high is the genus Lactobacillus. Again, your probiotic acting like an opportunistic pathogen. For more on the terrible nature of Lactobacillus, watch my webinar entitled Lactobacillus Probiotics, a Dumb Idea in Those Who Are Dysbiotic. And lastly, very consistently, 100% of the time, when a significant difference was found, Akkermansia mucinophylla was higher in dementia. If you don't believe me, I've even included the PMID numbers for you in the blue box. Mucinophylla may be a great idea for metabolic syndrome and IBD, but it is not a good idea for colorectal cancer, dementia, and Parkinson's disease. Here I compare the microbial signatures of dementia and Parkinson's disease. As you can see, they are quite similar. First, we see the superhero of the gut, F. Troussetii, almost always significantly higher in healthy controls, along with members from Lactobacillus, an amazingly beneficial family of genera. And Roseburia is one of those genera, and we also see Ruminococcus. We also see similarities in the bad actors, such as E. coli, Lactobacillus, and Mucinophylla. So this is concerning to see, because Parkinson's is a Lewy body disease, and there is Lewy body dementia. And dementia in general has a Lewy body connection. The data is 100% consistent, so I do not recommend the use of eosinophilus supplements in either of these two conditions, and to include colorectal cancer. If you want more information, watch my video on Akkermansia mucinophila and colorectal cancer. So what's going on with Akkermansia and Lewy bodies? Well, first, it's not the focus of this presentation. And second, this is literally the only paper I can find on the topic. So if you want, get this paper. So let's dive into this Lewy body topic and the connection between Parkinson's and dementia. So what is a Lewy body anyway? It's a clumping together of a usually normal component of neurons called alpha-synuclein. But in this case, these proteins are improperly folded, causing the aggregation. In Parkinson's, there are genetic mutations of alpha-synuclein conferring a higher risk, and the Lewy bodies are very abundant in the part of the brain most relevant in Parkinson's. In Alzheimer's, about half of those who died from the disease will also have Lewy bodies. And obviously, in Lewy body dementia, it plays an integral role. In fact, the distinction between Parkinson's and Lewy body dementia is made based on the onset of motor versus cognitive symptoms. Motor symptoms occur first in Parkinson's, cognitive symptoms occur first in Lewy body dementia. However, both can exhibit similar clinical symptoms as the disease progresses, with up to 83% of Parkinson's patients exhibiting dementia after 20 years. To refer back to apolipoprotein E at the beginning, ApoE4 carriers in Alzheimer's are more likely to have increased severity of Lewy body pathology. And in Parkinson's, ApoE4 was shown to increase the risk of Parkinson's disease dementia, as well as reduce the age of symptom onset. Lewy bodies don't just exist in the brain, but are found in other parts of the body, to include the intestines. In fact, the gut is where the origin of both diseases are theorized to originate. Parkinson's patients experience severe GI symptoms, primarily constipation, many years before the onset of motor symptoms. See my presentation on Parkinson's. Lewy body dementia patients also experience severe gut symptoms. So the two theories are as follows. Either LPS from dysbiosis causes alpha-synuclein aggregation, which has been shown to do, and this spreads up the vagal nerve into the brain, or dysbiosis in the gut causes permeability, and the high plasma levels of LPS cause brain permeability, allowing alpha-synuclein to enter and cause problems. This is confirmed in animal models, which have shown that alpha-synuclein injected into the intestinal wall could be transported via the vagal nerve to reach the brainstem. So either way, we need to address the gut microbiome. So let's try to put all of this together from the microbiome perspective. And although some patients with dementia don't have amyloid beta deposits, and in fewer cases, non-demented patients have as much plaque deposition as highly demented patients, we're going to stick with the conventional thinking on dementia disease progression. Clearly, not everything is understood. However, the role of inflammation is without question. Microbial data is very strong. The underlying question is, what is the root cause? What gets the ball rolling? If you have high blood sugar, that can be pro-inflammatory. But is that the root cause? In my opinion, there is plenty of data to suggest the root cause are microbes. And it's not just one microbe. There is no single smoking gun. There are plenty of opportunistic pathogens in our body to be the root cause. To me, it starts with dysbiosis. The two theories we mentioned earlier are alpha-synuclein slowly transiting the vagus nerve to the brain, as shown here, or LPS, whole bacteria, alpha-synuclein, and possible other components entering circulation and then enter the brain. So what happens in the brain, and who are the key players? Let's start at the beginning with the suspect number 1, E. coli. The whole bacterium or parts can enter the brain, as we've seen. We've also seen that the LPS on its outer membrane, shown here, is highly immunostimulatory. LPS is what's called a pathogen-associated molecular pattern. Our immune system doesn't have eyes and ears. Like us, if we see a bear, snake, tiger, or what have you, we go into fight or flight mode. Our immune system relies on chemical signatures, and E. coli LPS is a big one. The ever-present immune system recognizes the invader and mounts a defense. These primary players have names like astrocytes, beta-amyloid antimicrobial peptide, and microglia. The microglia are the resident macrophages in the CNS. They recognize LPS, and in an attempt to fight off the invader, inflammation results. Also fighting the invader are what are called beta-amyloid antimicrobial peptides, which act against fungi, viruses, and bacteria. Here is where we get into the amyloid plaque conversation. Beta-amyloid generation is an important immune pathway that mediates pathogen entrapment and protects against infection. After the killing off of the pathogen, the beta-amyloid deposit is normally cleared, but that appears to be defective in dementia. In turn, these deposits can then activate the microglia from earlier, leading to more inflammation. But why would this process stop working? One example to illustrate this is with ApoE4, which we covered earlier. The apolipoprotein E family, if you recall, activates the beta-amyloid peptides. But if this lipoprotein is defective, as is the case for ApoE4, then the risks for dementia skyrocket. Why? I don't think anyone knows for sure, but you can imagine that due to the fault within ApoE4, there was an improper signaling to the antimicrobial peptides, and they can't do their job well. Dysregulated antimicrobial peptide immunomodulation is a factor in the pathogenesis of a range of inflammation diseases, including atherosclerosis, lupus, arthritis, cystic fibrosis, asthma, psoriasis, rosacea, atopic dermatitis, and especially in Crohn's disease. See that presentation. And remember from earlier, a key point to keep in mind, for the genetic connection to Alzheimer's early in life, there are mutations in genes for encoding proteins for the amyloid beta. These beta-amyloid plaques are outside the cell. They then stimulate intracellular, inside the cell, neurofibrillary tangles, another term you might know. This is the classic signature of Alzheimer's, the accumulation of beta-amyloid containing extracellular plaques and tau containing intracellular neurofibrillary tangles. The aggregation of plaques and tangles is followed by microglia recruitment surrounding the plaques. This raises microglia activation and local inflammatory response, which advance the neurotoxicity. Reactive microglia activates astrocytes, another important glial cell that supports neuron functioning. Reactive astrocytes, as well as reactive microglia, contribute to neuroinflammatory burden. Now, normally, the microglia clean up the beta-amyloid deposits in a process called phagocytosis, but this appears to be broken. To make matters more complicated, a number of other bacteria produce amyloid. These amyloid proteins help the bacterial strains to form biofilms and to strongly bind to each other to resist destruction by physical and immune factors. Among those bacteria, guess who produces it? E. coli. The amyloids formed by bacteria are different from the CNS amyloids, but have similarities in structure. These similarities may trigger antigen cross-reactivity, which can trigger the immune system, which could lead to enhanced immune response with endogenous formation of neuronal amyloid in the brain. And the LPS-driven immune response with cross-reactivity can also occur in the gut, which brings us back to the original theory of dementia in Parkinson's via prions working their way up the vagus nerve. A prion is a misfolded protein that can induce misfolding of normal variants of the same protein and trigger cellular death. And like the beta-amyloid protein in Alzheimer's, in Parkinson's, the key feature, in this case alpha-synuclein, has antimicrobial properties, is regulated by ApoE4, causes microglia and astrocyte activation in the brain, is an abnormal aggregation of fibrils, Lewy bodies, and its aggregation is promoted by LPS. Sound familiar? I know this was a bit dense, but hopefully you can walk away with a better understanding of the origins of dementia. And there is no effective medicine. Clearly, your best bet is to address the root cause. Like we saw at the beginning, when dementia patients received a different microbiome, they were amazingly able to reverse their dementia. So what does that tell you? I'm not offering you FMT therapy, but if you think that's best, you can seek that out on your own. What I offer is the next best solution, that of dramatically altering the microbiome you have now, with the use of properly blended and dosed prebiotics to drive significant change for the better in your microbiome. I showed you earlier the microbial fingerprint for dementia. It was my meta-analysis of all the human fecal microbiome studies that I could find. There are clear differences between the microbiomes of the healthy versus those with dementia. There are significant reductions and amazing butyrate-producing superheroes inside the microbiome, and significant increases in bad actors like E. coli, which we mentioned a number of times. I know which prebiotics feed which bacteria. Visit my website, themicrobiomeexpert.com, which you can find a protocol for dementia. Get to the root cause. Start changing that microbiome. If you found this presentation informative, I have many more for free in my YouTube channel and also in the Microbiome University tab on my website, themicrobiomeexpert.com. There you can select from a wide variety of topics. And if you or a loved one are struggling with a disease/condition, I have condition-specific presentations as well, along with their microbiome protocols found within its respective tab on my website.